So let's start chatting about your ears. Okay, there are three parts to, if you consider an ear as a system, then there are three parts: the external, the middle, and the internal or the inner ear. So what are the parts of the external ear? First is auricle. You can see auricle or pinna right here. Auricle is a cartilage, more specifically elastic cartilage. It's covered by skin. Actually, it's one of the reasons why we can, partially the reason why we can, the pierced cartilage does not grow, does not heal back really well. So if the piercing goes through the cartilage, the hole is going to stay there, which is convenient. Okay, so the role of auricle or pinna is to direct the sound waves into the channel called external acoustic meatus, also known as auditory canal. So it consists of layer of elastic cartilage on the outside. So basically, like that. Sorry, that area, the more outer area, this area, and then on the inside, like this. This is the bone, the temporal bone of the skull. So those are plates of the cartilage, and this is the temporal bone. If you ever had the chance to take a look into the auditory canal of every person using the otoscope, you know that it's pretty nasty. There's a bunch of hair in everybody's ears. There are sebaceous glands that produce sebum, and there are ceruminous glands that are changed sweat glands producing earwax. Do you need to clear earwax? You think so? Yeah. What can be the smallest object that you are allowed to put in your ear? By doctors, huh? Say again? The smallest object that can go in your ear is your elbow. It means that nothing is supposed to go in your external acoustic meatus. Nothing goes in your auditory canal. You're not supposed to clean it. You're not supposed to dig there in there, recreationally. There's a problem, doctors will deal with it, but other than that, you're not supposed to do anything. Because, nope, absolutely not. It all should happen on its own. And it will clean out by itself. So it's pretty messy there. But there's an explanation to that, folks. The earwax is sticky. So the hair will trap dust and debris, whatever, and earwax makes sure that it does not move, does not get any further. All right, make sense? So once you go, and we literally going to be like, most of this conversation will be us. Imagine us turning into tiny little Oompa Loompas and walking through the ear system. So we walked through the external acoustic meatus, and now we arrived to the tympanic membrane. Tympanic membrane is the boundary between the outer and the middle ear. You also know this as an eardrum. Okay? It's translucent. Okay? So it's not transparent, but it's translucent, so light, you can kind of see light coming through. So the outer surface, this surface right here, the outer surface is covered by the skin, while the inner surface, so this is skin, and the inner surface here is mucosal membrane. Now the tympanic membrane is connected to auditory ossicles, 3 smallest bones in the human body. When the sound hits tympanic membrane, it vibrates, and when it vibrates, it in turn transfers those vibrations to these 3 auditory ossicles, malleus, incus, and stapes. So the sequence can be presented this way. Tympanic membrane, conveys the vibrations to the malleus, which conveys the vibrations to the incus, which conveys the vibrations to the stapes. Okay? So stapes in this illustration, it covers or sits on top of the so-called oval window. Which marks the start of the inner ear. Okay? Does that make sense? Awesome. Now, another opening that I want to highlight is the round window. And when I say a window, I want you to understand, it's, in case of oval window, it's not an opening. It's not an opening in the sense that it's like a hole. It's a hole that is covered by mucosal membrane. But it's not like an actual hole, like I don't know, your eye socket. Okay? So, oval window marks the beginning of the inner ear. Round window is what essentially is the end of an inner ear. But it's too early to talk about that, so let's focus on what we've got here. So a couple more anatomical details. First, pharyngotympanic tube. Pharyngotympanic tube connects middle ear and your pharynx. What is the non-fancy name for pharynx? Louder. Throat. So it's a connection between your throat and your middle ear. Okay? Normally, this tube, as you can see here, it's collapsed. When you yawn, it opens. Okay? And the point is to equalize the pressure in the middle ear and the environment. Does that make sense? For instance, when you are on a plane flying, do you have the, I don't know the proper English word for it. When your ears are like, you are popping them by doing this, right? By increasing the pressure inside of your throat and in your inner ear. Or another option, sometimes it works, you just open your mouth and they pop, right? That can happen too. Am I making sense right now? Yes. So this connection between the throat and, so throat is here. That way. So that connection creates a lot of problems in kids, because kids tend to have a lot of nasal infections. And when they have nasal infections, there's a snot running in the back wall of their throats. So if it's a kid, the auditory canal is shorter and more horizontal. So it's easier for those bacteria to get into the inner ear and cause otitis media, accumulation of the pus in the middle ear. So this is what a healthy eardrum and the eardrum with otitis media, what they look like. You can see how reddened it is because it's inflamed. And this is a leading cause, one of the leading causes of hearing loss in children. Well, with age, definitely hearing loss. And you see this rupture of the eardrum. So I want to clarify, folks. Treatment for this condition is myringotomy. Okay, this insertion of the tubes, yeah. So obviously, if you put the tubes, you have to make a hole. And when they come out, this hole heals. So it's not like the rupture, the damage to the eardrum is irreversible. But if it's big enough, if it's like shattered, even if it heals, there will be scar tissue formed. Scar tissue makes it less elastic, which means it's going to fail to properly vibrate. Am I making sense? Now, I don't know how interesting the functional anatomy for you. I totally get if it's not. But here's a little clinical relevance. Let's say your child, because I want you to try the role of parents. Probably many of you are parents. Your child gets an ear infection. Okay? Complain that it hurts. And it's Monday. What do you do? Say again? Take him to the doctor. Yes. So the doctor can do what? There you go. Yes. Take a look at the ear and give him antibiotics. So that was a really good clinical study, fantastic clinical study, because I'm a big proponent of evidence-based medicine. They decided to look at kids with ear infection and see what difference do antibiotics make. Okay? It's called, technically for this, it's not even non-inferiority. So they had a group of kids that were told not to do anything. Just nothing. Maybe they were given like saline or something. And then a group of kids with their infections received proper antibiotics. And they looked at the full recovery or like disappearance of pain, something like that. I think disappearance of pain. 3rd or 5th day after the start. So if it's Monday, right, it's going to be like Wednesday. They looked at Wednesday or Friday, whatever. The point is, certain day after the symptoms occurred. And antibiotics caused the resolution of symptoms in 89% of children. Good. Group with no antibiotics showed resolution of symptoms by the same date in 85% kids. So there's 4% difference, which is not huge. So now I think it's American College of Pediatrics. They recommend when there's a child with ear infection, the doctor either, now it's all electronic, so sends an electronic prescription to the pharmacy and puts like do not feel yet, like delayed, okay? If a child feels better, it's not needed. If a child still feels crummy, parents can call the pharmacy or call the doctor and they will activate. And they can pick up antibiotics and give it because the infection is worse. Does that make sense? That's the only way, yeah. Okay? Yeah. So if everything's good, you don't need them. Now, these three bones, the incus, the malleus, the incus, and the stapes are connected to each other. Okay, malleus is attached to the eardrum, stapes is to the oval window, and now imagine you are hearing the sound, right? Sound is vibrations of air, so sound vibrates the tympanic membrane. The tympanic membrane vibrates all ossicles, and the waves go into your inner ear. Make sense? What if the sound is super, super loud? Will vibrations have larger or smaller amplitude? Will they be more sort of I'm losing words. More vigorous or less vigorous? What do you think? Loud sound. More vigorous, right? So it will be like super back there and back. Now, if you're going to be rattling those three bones at a great amplitude, can it potentially damage them? So you want to limit the range of the vibrations, the amplitude, right? It is achieved by two muscles. They're not in the on the picture, but tensor tympani is attached to the malleus. Stapedius, or stapes, is attached. Stapedius muscle is attached to stapes. Sorry, long day, I'm misspeaking. When the sound becomes very loud, these muscles become stiff, okay? And when they become stiff, these bones cannot move as vigorously as they did before the stiffening of those muscles. Am I making sense? So basically imagine that I do this with my arm and somebody comes over and grabs my arm. I can move, but my movements are now limited. Okay? These muscles do not relax quickly when they stiffen. So if you've ever been to a really loud concert, really loud, when you go out of that concert, you're a little bit deaf. I went to a Napalm Death concert once, still questioning why, and I was in the second row of one of my favorite bands, Dream Theater. Second row. They were, guitar player was as close to me as Shanat, like that close. Next day I had an NP1 class, and I yelled at them because I could not hear myself. And I could not hear myself a few more days. Sure.
So they have the things that you can put in your ears that you're supposed to
For
how much it hurts.
this.
Sound about, but it doesn't
This.
temporary.
Tubes. So tubes. These ones. When people say put a tube in the ear,
they're bigger. They're almost like earbuds, but you can still hear. People wear them at concerts because it's supposed to fit.
oh, you're talking about earplugs.
Yeah.
Yeah, they just obstruct the acoustic meatus, and sound just doesn't reach well. That's it.
Because these ones, like, you can still hear it, but it's like...
Oh yeah, in a good concert, they're not going to help. Like, well, they will, but you will still hear, trust me. I know. I saw him. Super excited. Okay. Now, this is, okay. I kind of hate it because there are a lot of details. So let me highlight the most important parts, okay? So we're going to start, and I'm going to use some props here. We're going to start with two types of labyrinths, bony and membranous. Now, here's what I want you to appreciate. So imagine you take human head, okay, and you pour human skull. All the non-bony tissues are gone. So when you start cutting human skull in the area of inner ear, you're going to see a cavity. That cavity within the bone is called a bony labyrinth. Am I making sense? When you look right here, these are the walls of the bone. So what you see is the shape of a carving within the bone. Bony labyrinth isn't something that you can cut out of the human skull. It's like an eye socket. Does that make sense? Like you can look at the skull and say this empty space is an eye socket, but you don't cut out a part of bone and says it's an eye socket. It's an empty space. Is that clear? So bony labyrinth is exactly this, an empty space. This empty space is filled with the liquid called perilymph, which is very similar to the cerebrospinal fluid. The parts of the bony labyrinth are semicircular canals. Okay, I'm gonna keep assigning the roles for them so you know what they're for. So semicircular canals, dynamic balance or rotation, vestibule, which is static balance or linear movement, and cochlea, which is structure responsible for hearing. Remember, all those elements of bony labyrinth are cavities. Within these cavities you can find inside semicircular canals you will find semicircular ducts. In the vestibule there will be utricle and saccule, and in the cochlea there will be cochlear duct. Now here's my question, and you don't really need to be a specialist in any kind of biology or medicine. When we use the word duct, what does it imply? Like, is there something inside of that duct? Yes. Sorry, I accidentally clicked. Yes, there we go. Okay, so there's something inside of that duct. So let's figure out what's inside. Within these ducts that are in a blue color here, so this is, for example, a cochlear duct. Within those ducts also a fluid, okay, which is called an endolymph. Does that make sense? So the illustration that I wanted to provide for you, I wanted to draw something like this. So it's a generic image, okay, folks? This is bone. This empty space within the bone is the bony labyrinth. Okay? This bony labyrinth is filled with perilymph, and within that perilymph you have an actual membranous labyrinth. Okay, so these are membranes, call them walls. This is the membranous labyrinth, and inside it contains the fluid called endolymph, which contains a lot of potassium. Does that make sense here? Okay? So far. Now, in our conversation, there are two really important considerations you have to give it. One is the fact that both perilymph and endolymph are fluids. Two, when we get to hearing, especially, is the fact that endolymph contains a lot of potassium, which is quite unusual because, let me ask you this. If we compare the environment inside the cell and outside of the cell, where do you find more potassium? Inside of the cell or outside? Inside. This is the only case where it's opposite. There's so much potassium that the concentration of potassium in endolymph is greater than the concentration of potassium inside the cell. We're going to keep it in mind for now. Okay? Does that make sense? Now, obviously, this system, the inner ear, is connected to the brain because this is where it transmits signals to by vestibulocochlear nerves, and technically it's several nerves. So this is the cochlear nerve. That's hearing, and that's vestibular. Balance. Right now we don't really need to talk about that distinction. So now let's talk about cochlea. Hear me out. I went absolutely bananas on the cochlea, on the anatomical details. So what I'm going to do, I'm going to give you the overview of cochlear anatomy using both this image on the right and my drawings. But before we get there, I want to go back and ask you to look at the cochlea one more time. The cochlear duct, more specific, and answer my simple question. Does it look a little bit like a cinnamon roll? If you take a cinnamon roll, hypothetically, hear me out, it's important. When you take a cinnamon roll, can you sort of unroll it and sort of straighten it up? This is what we're going to do with cochlea right now. In our minds, okay? What we're going to do is uncoiling it and stretching it out. So when we do this, we get something like this. It's a very short cochlea, I must admit, but something like this. Okay, good? So this is cochlear. This is cochlea, the shape. Okay, within that shape, we have that cochlear duct, which also straightened out. Now imagine I'm going to milk my cinnamon roll analogy. Now you have that cinnamon roll straightened out. You with me? Now take a knife and cut it along. Can you do that? Hypothetically. Yes. This is exactly what we're going to do. We're going to cut it along. And what you're looking at here is the longitudinal section, schematic longitudinal section of the cochlea. So this here is the oval window. Remember, there is a bone called stapes, and stapes will vibrate when there is a sound. Am I making sense, folks? So these vibrations will be transferred through the oval window within the cochlea. What feels that part? What is here? Perilymph, right? Perilymph is a fluid. If somebody throws a grenade at you and it's about to land in the water and you decide to jump in the water, what's going to happen to you? Huh? Yep. You're going to get murdered. You're going to get killed because vibrations in water travel farther than vibrations in the air. Sound in water travels farther. Water is much better conductor of sound. We doing all right? Excellent. So here are your vibrations. Now this part is the cochlear duct. Is it filled with fluid? What is the name of that fluid? If the sound is in the proper range, it will cut through endolymph. It will make endolymph vibrate, and these vibrations will trigger organ of Corti, or spiral organ, which then will convert the vibrations into the nerve impulses that you perceive as a sound. Does that make sense? I'm going to do a run through of all this. And then those vibrations, remember still, that's a movement, that's a mechanical energy. It has to escape somewhere. So they get out through perilymph again, okay, and dissipate through the round window. Is that clear? Does that make sense? Now, you may ask, what if the vibrations, the sound is not in the hearing range? Let's say it's infrasound or ultrasound. It will not trigger the vibrations of endolymph and just circle around. So finally, we need to name all those compartments. This is 1, this is 2, and this is 3. So 1 is the scala, vestibuli, 2 is scala media, also known as cochlear duct. And 3 is scala tympani. Sorry. Let me know when I can move back. Right? So now, right now, just ignore the text. Okay? What you see here in this image is not a longitudinal, but a cross section. So the top channel is, let me make some connections here. Scala vestibuli. This is scala tympani. And this is scala media. And this is your organ of Corti. Does that make sense? Now they are separated by the membranes. So if we continue working on this, it's a bit overloaded, I know. I'm fully aware of that. But a couple more. They're membranes. So this is the vestibular membrane. And this is the basilar membrane. So, question, what do you need to know here? For the sake of brevity, I'm just going to highlight elements of functional anatomy that I can totally ask you about. If I don't highlight something, I will not ask about that. So, absolutely know what the organ of Corti is, what is it for, where it is, and be able to identify it on that image that you see. Scala vestibuli and scala tympani, yes, absolutely, 100%. Okay? Know that both of them are parts of the bony labyrinth, okay? And know that scala vestibuli starts at the oval window, scala tympani stops at the round window. Scala media, yes, cochlear duct contains endolymph. What they are separated, look. Scala media and scala vestibuli separated by the vestibular membrane. Scala media and scala tympani by the basilar membrane. Okay? Now, to finish this, actually, you know what? Let's stop right there. I will explain why. That we're going to answer is how the sound itself is converted into electrical impulses in your neurons, in your nerves. Okay? So, for this, first we're going to discuss what the sound is physically. So, sound is the waves. Okay, you can see here that when something produces the sound, it produces the areas of more compressed air and the areas of less compressed air that can be perceived as waves. So every sound that we hear has a certain frequency, which is a pitch. Okay? And it also has an amplitude, which is the loudness. So those are two main qualities of a sound, and we're going to discuss how we can distinguish sounds of different amplitude, different loudness, and different frequency or different pitch. So the unit to measure frequency is hertz. Hertz in general is the unit that shows you that there is one wave per second. So human ear can hear sounds in the range from 20 hertz to 2000 hertz. Please do not memorize you can memorize the numbers, but I'm not going to ask those numbers on the exam. Not the entire range. We cannot hear entire range equally well. There is a range about, I think, 100 to 4,000 hertz. That is like the best hearing. Okay? So in the area of 1,500 to 4,000 hertz, we can distinguish frequencies that are very close to each other. So what does that mean? Are you familiar with the perfect pitch? So I can assure you that every person in this room can hear the differences in different pitches at the same level. Do you understand what I'm saying? So like, if my voice modulation may be subtle, but you can hear them anyway, it is whether or not you can identify those frequencies, identify those pitches, and tell me, oh, this is the A of the first octave note, or this is the B of the second octave, and so on and so forth. Perfect pitch can be trained, but it is a part of sort of intrinsic capabilities. Pretty much the same way, like if you think about people that are really good at something, okay, for instance, people who do the, coffee, like tasting, professional tasters. We can taste the same stuff they can, but we can't really articulate what we are tasting. We can't give feedback. The same way, the person with the perfect pitch, you play them a melody, and they play it back to you because they can match what they hear with the keyboard or strings or whatever, frets, right? And everybody else just can't. Now, amplitude is measured in very weird units, which are decibels. Why I say that it's weird units? Because every time you add 10 more decibels, you increase the loudness of sound 10 times. So it's a weird scale, okay? Let me give you some examples. This is one of my favorites. So first of all, if you look at this interesting graph, it shows you the range of hearing of different animals. So these are humans, and we have pretty amazing hearing, range-wise. The only animals that are better are whales and dolphins, which makes sense, because that's how they communicate. Again, you start looking at, different animals, and sometimes it kind of makes sense. So, for instance, you can see that bats, crickets, and moths all can hear in what we consider ultrasound, because they use it for location. So for bats, it's important to locate the prey, and for insects that are crickets and moths, it's important to avoid being eaten. Okay? Now, if you look at the comparison of different frequencies right here, you can see how different sounds are distributed. So, like vowels have a lower pitch, consonants, the, what's it called? Like a hissing consonants, higher pitch. the important sounds all fall in this range. This is where our hearing is ideal. Not ideal, but the best. Now, this is just an example of the frequency range, different musical instruments and human voices. So, female voices, on average, higher. Violin, trombone, flute, all fall into that relatively low frequency, well, relatively compared to 20,000 frequency. Now, this is my favorite because it talks about loudness. So, normal breathing is 10 decibels, which is very quiet. Rustling leaves is 20, which means, hear me out, it's 10 times louder than breathing. So every time you add 10, it's 10 times more. Okay, but I want to skip the middle and get to these extremes. Okay, are you familiar with, well, first of all, a lot of records that humans set are rather stupid. Have you heard about the record for the loudest crowd noise in the football stadium? Several years ago, well, very several years ago, pre-pandemic, this record was set at the Seattle Seahawks stadium. It was designed, people brought some noise-making devices, like they put earplugs and everything. The sound was recorded in 132 decibels, which puts it slightly above the machine gun loudness. Okay? Which is very interesting, but I don't know if you're familiar, there is a world record in the loudest ever recorded snore. And I want you to guess where on this scale is the snore. The record still holds. Give me like between what and what? You under 110, you underestimate human beings. The loudest snore ever recorded was 127 decibels, which puts it above the threshold of pain. It's a male. Go ahead. Question?
So what is the threshold of pain? What does that mean?
Like it hurts.
Okay,
Like your eardrum, the tympanic membrane, is at a very good risk of just popping because of how loud the sound is.
Do we have to do like a sleep apnea?
I don't know what what I want to highlight, it's not like that this person was snoring like that every night, but he was a very loud snorer, and they put the recording equipment, and people recorded the loudest snore. It's a male. At the time of recording, at least, he was married. His wife is deaf on one ear, which is not surprising. Yeah. So, now we're going to go and figure out how sound is converted into electrical impulses. And we're going to start with reminding ourselves how sound gets into the inner ear, into the cochlea. So remember, sound first is focused by the auricle or pinna, the external ear, into the external acoustic meatus or auditory canal, and eventually the sound reaches the tympanic membrane. Tympanic membrane vibrates with the same frequency as the sound has. These vibrations are transferred onto the auditory ossicles, malleus, incus, and eventually stapes. When you look at the relative size of the tympanic membrane and the oval window, see those sizes? Oval window is much smaller. So all that energy of the sound, all that pressure is transferred from the larger tympanic membrane onto a smaller oval window, which amplifies the sound, makes it louder, even louder. And then the sound enters the inner ear, which you may remember is filled with fluid. So these vibrations transmitted to the perilymph in the scala vestibuli, and if these vibrations are in the range that we can hear, they will stimulate vibrations of the scala media, kind of cut across. Does that make sense? And then eventually they will cross scala media, enter scala tympani, and will be released, the energy will be released through the round window. If the sound is not in your hearing range, it will not excite your scala media and will just go around, go into scala tympani and will leave. Okay? That makes sense? So, when we say that we can't hear certain sounds, like we can't hear ultrasound, or we can't hear infrasound, super low sounds, does that mean some of them, all of them, have no effect on us? Huh? Well, for ultrasound, we really don't know, and apparently they don't. But infrasounds, like really, really low sounds, like 3 hertz, have a very interesting effect on human beings. If I would bring infrasound generator here and turn it on, we all, without an exception, would start panicking, just rush out of the room. Infrasounds scare humans. We experience unexplained, irrational fear when we hear them. Nobody knows why, but it's a known physiological phenomenon. Okay? Does that make sense? So, this is basically the summary of what I just told you, okay, in the form of flowchart. I'm not going to read it again to you. Like, seriously, it's exactly what I just told you. Okay, we're good? And I'm going to move on to the spiral organ, or organ of Corti, or hearing organ. So what you need to know. So this is the spiral organ. It is positioned on the basilar membrane. Basilar membrane separates scala tympani, which contains perilymph, so it's under, and scala media, which is here, that contains endolymph. The cells that we are going to focus our attention right now are the inner hair cells, these little guys. Okay? So if you look very, very close at a hair cell, you can see that it has this hair. Right? The roots of, and I'm going to put this in the parentheses because it's not really hair. So the roots are in the hair cell, and the tips are in the tictorial membrane. Okay? Wait a second. So as I said, before the system shut me down, the hair cells are confined between the tictorial membrane and the inner hair cell. So what are those hairs? You can see the inner hair cells. So these images show you how the hair cells are looking like. Take a look at this. So these are the auditory hair. Okay? These auditory hair are effectively cilia, so-called stereocilia, and the largest of them is kinocilia. So far, we good? Now, if you look at the electron micrograph on the right, you can see that these individual cilia are connected to each other. Can you see the links? Can you see the links? Those are tip links. So why this is important? Hear me out. If one kinocilium, for example, if it bends, it's going to bend with it all other stereocilia. Do you agree with that statement? Because of the links. Like imagine we all are cilia, right? We're all holding hands. And we're standing upright. And I start falling down. I will pull all of you, directly or indirectly with me, into the same general direction. Is that good? Understood? So now, before we move on, we need to summarize this image. So there are inner hair cells, which stereocilia are held tightly by the tictorial membrane. Stereocilia are connected to each other, and when one of them bends, others will bend as well. And finally, the important part is that these inner hair cells are within the scala media, within the endolymph. And remember that endolymph is really rich in potassium. So now that we are equipped with that knowledge, let's figure out how the cells convert vibrations into an electrical signal. So to answer this question, I first have to go back to this image and remind you what is where. So this part, the scala vestibuli, contains perilymph, as well as scala tympani. Basically, cerebrospinal fluid. But scala media contains endolymph, which is potassium rich. Your hearing organ, the organ of Corti, is here, sitting on the basilar membrane. Now look what happens when there are vibrations. Check it out. You see that the basilar membrane vibrates as the sound crosses through scala media. Making sense? Yeah? So now let's go back to this and see what happens when the sound is arriving to your inner ear. This image here represents, there's no sound. Make sense? No sound. And then there is a sound. And because of those vibrations of the basilar membrane, these stereocilia bend. Make sense? When they bend, they open potassium channels. Since endolymph is rich in potassium, potassium diffuses into the hair cell. What is the charge on the potassium? When you bring positive charges in the cell, what happens to its membrane potential? It becomes more positive, and what do we call it? Depolarization. So cell becomes depolarized. What type of channels does change in voltage open? How are they gated? I gave you an answer in the question. Voltage gated, yes. So the depolarization opens voltage gated calcium channels. For some reason they aren't here. And voltage gated calcium channels allow calcium in, which stimulates the release of neurotransmitter. Okay? Does that make sense? So now, take a look at the same process. I'm just going to highlight how to go about this process when you look at the text. So, basilar membrane moves in the response to sound. All good? That movement bends stereocilia. All good? That bending opens potassium channels. Potassium diffuses into the hair cell. So is calcium. Cell becomes depolarized. More intracellular calcium means the release of neurotransmitter. The release of neurotransmitter increases the frequency of action potentials that are formed by the neurons in the cochlear nerve, and you hear a sound. Clear? So while we are at this, how do you distinguish sounds of different loudness? Think about what loudness is. It's the amplitude of vibrations. If the vibrations of the basilar membrane have a greater amplitude, it means that these channels stay open longer, which means more potassium and calcium diffuse into cells, which means calcium stays up for an extended period of time, which means more neurotransmitter is released, which means the frequency of action potentials increases even more. So the more frequent the action potentials are, the louder the sound. Sounds good? Now, how do we know the pitch? So this is the lovely story, and mostly pay attention to the picture. So this is the sound. This is a very, very high frequency sound. Okay? So it's going to vibrate a very specific set of fibers in the basilar membrane. So like this, the short fibers. Okay? The short fibers will respond only to high frequency vibrations. And only the inner hair cells that are located here will transmit the signal to the brain. And it's going to be an individual pathway that gets into your brain and tells it those specific fibers were vibrating. I'm going to give you a pretty good analogy. Hear me out. Okay? If the sound becomes lower, it will vibrate the part of the basilar membrane that is longer and not as stiff. So, anyone here, did you ever look inside the grand piano? All those strings have different length. And that, in part, not only this, but that's in part what determines the pitch for every key in the piano, because it hits an individual string. That makes sense? This is in reverse. When a specific quote-unquote string, the fiber of basilar membrane, when a certain string vibrates, it can only vibrate if it's stimulated by a particular sound. Now, you may ask, what if we hear a chord? Well, different notes will vibrate different fibers at the same time, and your brain will receive signals from different fibers at the same time and will perceive it as a polyphonic sound, a chord. Does it make sense? Okay, so we figured out amplitude, we figured out frequency, and obviously if you have a category of cells that are called inner hair cells, there's got to be outer, and indeed there are. So, what are they for? So to explain this, I'm going to give you an explanation in the video, then I'm going to pause it and show it to you using my hands. So, when the sound that you hear is very loud, your brain sends back the signal to the outer hair cells, and they stiffen in response to that signal from the brain and limit the motions of the basilar membrane. Now, in terms of the pathway, again, I'm not asking you about all the different nuclei or whatever. So you have the hearing organ, okay? And the signal gets in the medulla and midbrain, and then the thalamus. Now, let me ask you this. Can you think of a reflex, automatic behavior that is associated with a sound? Oh, there you go. That's the reflex. Startle reflex, right? Startle reflex. So this is why midbrain. Midbrain is responsible for startle reflex. But your actual processing, so thalamus, will direct the sounds to various regions, mostly to the auditory cortices in the temporal region, okay? And the primary auditory cortex will interpret the pitch. So which parts of the spiral organ specifically send you the signals. It will determine the loudness. Okay? So far. Doing all right? And it will determine the location of the sound. Okay? Now, how does it determine the location? Turns out, when I'm talking to you, usually one of your ears is closer to the sound source than another. It means that the closer ear receives the sounds first. So there's a bit of a timing discrepancy, and the sound is slightly louder in the closer ear. Based on those very, very tiny little differences, the brain can interpret where the sound is coming from. And people who are deaf on one ear have a bit of a trouble determining the location of the sound compared to people who hear with both ears normally. Does that make sense? So, pathologies. Well, deafness. There's nothing else to say. But there are two types of deafness. There's a conduction, and there's sensorineural. Conduction deafness. Anything that happens in your outer or middle ear. Your tympanic membrane is completely trashed. Your auditory tube is blocked by something. Effectively, when you, mow the lawn and you put the earmuffs, yeah, that's effectively conduction deafness. Okay? It can be treated. Oh, another interesting, otosclerosis, which is really cool. Ossicles confused to each other. They have two joints, and now they don't have joints. And that's not good. Can be fixed surgically. Okay? Yeah, bones are fusing to each other. In case of sensorineural deafness, though, the damage is basically to the inner ear. So, first of all, older people, do they have better or worse? Worse. As you get older, you lose inner hair cells. When you constantly listen to loud noise all the time, all those vibrations progressively destroy those hair cells. Okay? And you get deaf. I don't know how many of you are familiar with the band called AC/DC, but their lead singer, Brian Johnson, had to retire several years ago because he was explicitly told, one more show, you're deaf. Okay? I don't know how many of you are familiar with Ted Nugent, but this dude is indeed deaf. On one ear, he boasts how loud his concerts are. Okay? I know. Now, what else can impair the hearing? Infections. It's a known fact that some, interestingly enough, respiratory infections can result in the damage to the cochlear nerve and lead to deafness. In our family, my mom has been affected by that, and my brother. They both have poor hearing in one ear, both after influenza. So get your vaccine. More seriously, tumors. I mean, obviously, if somebody has a malignant brain tumor, deafness is probably at the very bottom of the list of the worries. But sometimes tumors can be very small and benign. And if a tumor starts pressing on the cochlear branch of the vestibulocochlear nerve, it will cause a hearing impairment. Okay? Now, it can be helped. Cochlear implants can help with that. Okay? Now, I can't not put a plug for my favorite viruses in this. There's a virus called cytomegalovirus. You may have heard about it, may have not. It's actually absolutely benign, pretty much benign in adults. But if it infects a child in utero, it can come across placenta, you know. If it does that, the best outcome for that child is to be born deaf, because all other consequences are much worse. It's a leading cause of congenital deafness in newborns. The infection with cytomegalovirus during pregnancy. Tinnitus, ear ringing, auditory hallucinations. Nothing bad about it. It's a sign that your hearing is getting worse and worse. Okay? Just ringing in the ear. Good? So those are main problems with the ear. We're going to take a little pause.